I am Mrs. Thelma Pilin, your facilitator this afternoon. As given two topics, the specialized connective tissues, cartilage, and bone. But before we go into the bone tissues, we'll start first with cartilage. And I would like to have a little bit of a brief review on the formation of connective tissues in the body. So we'll have a brief review on histogenesis. There are two cells involved here, the egg cell and the sperm cell. The egg cell could be round in shape, could be ovoid in shape, and aside from the cell membrane, it is protected by two layers. We have the corona radiata and zona pellucida. The corona radiata is made up of follicular cells forming a crown around the egg cell. And next to that is a translucent layer we call the zona pellucida. The sperm cell, they are tadpole in appearance, composing of three parts. We have the head portion, the middle portion or the midpiece, and the tail portion. Now, they swim towards the egg cell during fertilization by using their tail with the characteristic lashing movement or whip-like movement of the tail. Now, the first sperm cell that touches the corona radiata will open up the cap-like structure in the acrosome or in the head portion and will release an enzyme that will destroy the follicular cells of the corona radiata. Other sperm cells that reaches the egg cell will just help the first sperm cell to destroy the outer layer because the first sperm cell that touches the corona radiata will create a reaction within the egg cell we call the zona reaction, meaning the egg cell will be impermeable to other sperm cell. It will only entertain the first sperm cell that have touched the corona radiata. The egg cell contains a pronucleus containing 23 chromosomes. Likewise, the sperm cell contains a pronucleus containing 23 chromosomes. So during fertilization or during the union of the egg cell and the sperm cell, the chromosomal numbers of both the sperm and the egg cell will become complete. So from haploid to diploid. Another structure or organ involved in fertilization, we have the uterus, especially the fallopian tube of the uterus. So the uterus is a pear-shaped organ having a uterine tube on both sides of the organ. The uterine tube or fallopian tube is composed of interstitial part wherein we have there the isthmus or the neck portion of the uterine tube. We have the ampulla and we have the infundibulum and fimbria or fimbriae. Now the fimbriae has at the edge portion of the structure we have a finger-like projection like fingers, no? And their function is to catch the egg cell during ovulation period. And the egg cell will migrate towards the ampulla or the ampullary region of the uterine tube, wherein this is the site of fertilization. We have here an illustration of the uterus with the uterine tube and the ovary. The egg cell is developed in the ovary. So if the follicle within the ovary reaches the ripe stage, which we call the Graafian follicle, it's now the time that the egg in the Graafian follicle will be released, and this is during ovulation period. So the fimbriae of the uterine tube will catch the egg, will reach the gastrula stage. And in the gastrula stage, the trilaminar germ disc is formed, and from this trilaminar germ disc, the three germ layers are developed. We have the ectoderm, we have the endoderm, and we have the mesoderm. The mesoderm have a great role in the development of the connective tissues of the body. So during the time that the cells will be developing or will be differentiating into connective tissue, the first tissue, or we call the precursor connective tissue of the body, is formed, and we call this as the mesenchyme. Okay, so this is the second stage of cytomorphosis of the cell. From the time that the cell is formed, we have the embryonal stage, and the second stage is differentiation and specialization until it will reach its maturity. This is the illustration of the mesenchyme, the precursor of connective tissue in the body. So this is the youngest type of connective tissue in the body. So this slide is a fetal kidney wherein this has been stained using the H&E staining technique or the hematoxylin and eosin technique. So embedded in the mesenchyme will be the collagen fibers. And you can see the mesenchymal cells, which we call here undifferentiated mesenchymal, to be differentiated into cartilage cell. So the mesenchymal cells or the undifferentiated mesenchymal cells have a characteristic cytoplasmic extension within the cell, and they are actually interconnected with the other mesenchymal cells, forming a network. And the ICS is gel-like in nature. So the mesenchyme is the youngest connective tissue of the body, or we call the precursor of the connective tissue of the body, and will develop into connective tissue proper and specialized connective tissue. So connective tissue proper, we have the loose connective tissue, we have the dense connective tissue, we have the adipose tissue, and specialized connective tissue, we have the vascular, we have the cartilage, and the bone. I want you to be aware of the general characteristics of connective tissues of the body, so that we will understand better why we have this specialized type of connective tissue. For the connective tissue proper, we have four general characteristics. One, the connective tissue have fewer cells that are set widely apart from each other. They have an abundant amorphous ground substance or intercellular substance, and the connective tissue fibers are embedded in the AGS, or in the amorphous ground substance. And take note for number 4, the connective tissue proper are highly vascularized except the mucous connective tissue and cartilage. Let's go into the specialized connective tissue, the cartilage, and the bone. We'll start first with the cartilage. Why are they classified as specialized connective tissue of the body? Okay, these two tissues, the cartilage and the bone. By knowing the characteristics of the connective tissue proper and comparing it to the characteristics of the cartilage and the bone tissue, we can understand better why cartilage and bone tissue are considered to be a specialized type of connective tissue. So in the connective tissue proper, the ICS, or the amorphous ground substance, is gel-like matrix, okay, and they are highly vascularized. While in cartilage, the ICS is soft but flexible gel-like matrix, and they are avascular. The bone tissue have a solid matrix or hard ICS and is highly vascularized. So we can see the differences in the characteristics of this tissue. We have the connective tissue proper, the cartilage, and the bone tissue. We'll start first with the cartilage. The cartilage is described to be a dense, firm, but pliable connective tissue. It is composed of cartilage cells we call the chondrocytes and an ICS that is gel-like in nature but flexible. Now, embedded in the ICS are the fibers. We have the collagen fibers and elastic fibers. So if you will go into the general characteristics of cartilage, they have an abundant chondrocytes as a rule, presence of connective tissue fibers. The type will be depending on the kind of cartilage involved, solid but pliable ICS, and is avascular. So take note of these four characteristics of the cartilage. Let's go into the cartilage formation. During the second stage of cytomorphosis, wherein the cells will undergo, or the undifferentiated mesenchymal cells will undergo differentiation and maturation, or they will be developed to form a cartilage in the body, the mesenchymal cells loses its cytoplasmic extension. So they will be grouped together and will become ovoid or oblong in shape. And later on, through the deposition of the matrix, the cells will be set farther apart from each other, and some of them will be trapped in a cavity, or we call the lacuna or lacunae. No? So in each cavity, it could contain one chondrocyte or maybe two chondrocytes, a maximum of 10 chondrocytes. And if ever they are formed, we call them as the isogenous groups or cells. During the time that the chondrocytes are trapped in a cavity, under the microscope, it will give us a different staining capacity of the different areas. So just above the cavity, that area there will be darker in color. We call this area as territorial matrix, and they have a darker appearance under the microscope because there are more of the acid mucopolysaccharide present in that area. So they are deeply basophilic areas. Now, further apart from the cavity in between the chondrocytes or in between isogenous groups, there is a lighter staining area we call the interstitial matrix or interterritorial matrix, and they will appear pale under the microscope because they are less basophilic areas. So we have here a closer look of a chondrocyte within a lacuna. So we have the cavity here, which we call the lacuna, and each lacuna may contain one or maybe more than one chondrocyte. So embedded also in the ICS are the collagen fibers, or in the round substance, or in the ICS. So we have the cells in a lacuna. From a young cell, we have the chondroblast, which will mature later on to become chondrocytes. So chondrocytes are the mature cartilage cells. Now, the chondrocytes, they differ in shape based on their location. When they are still young cells, they have an elliptic shape and will change to round cells, maybe to round or ovoid cells when they become mature, ranging from 10 to 30 micrometer in diameter. If they are trapped within a lacuna may contain 2 or 10 chondroblast or chondrocytes. And if ever they are trapped together inside a lacuna, we call them either as isogenous groups or cell nest or cell families. So the function of the chondrocytes from chondroblast to chondrocytes is to synthesize and secrete the extracellular matrix or the ICS, and they surround the cells within the lacuna. The cartilage matrix is now the secretory product of the cells, and its characteristic is that it is homogeneous in nature. It appears like a transparent substance, a gel-like transparent substance. It's because the tropocollagen or the collagen fibers may have the same refractive index as that of the matrix. So they will appear as transparent or homogeneous in the cartilage matrix is principally composed of proteoglycans, a combination of protein and carbohydrates, so which is commonly referred to as glycosaminoglycans or GAGs. So under the microscope, the morphology of the matrix will appear to be like a bristle of a feeding bottle brush. So the core, no, the core part of the chemical substance will be protein in nature, and the bristles the will be the gags made up of chondroitin sulfate and keratan sulfate. Then aside from the gags, we have the hyaluronic acid, which acts as a binder. So they act as a sort of cement to bind the proteoglycans together to form large aggregates. Okay, so we have hyaluronic acid and the gags. Let's now go into the perichondrium, which is a layer of dense connective tissue that is actually useful in maintaining the growth of the cartilage. This perichondrium is present in all hyaline cartilage except in articular cartilage of joints, especially in long bones of the body. And the perichondrium is rich in type 1 collagen fibers containing numerous fibroblasts. So we have here an illustration of a developing cartilage. So you can see on both sides we have the perichondrium. The perichondrium on the outer surface protects the chondrocytes in the middle portion, while the chondrocyte is undergoing deposition or setting the cells setting further apart from each other. Now, the perichondrium has an important characteristics we call chondrogenic layer. So, what is present in the perichondrium are young cells, are young cartilage cells. We have the chondroblast, which will later on mature to become chondrocytes and function to secrete the matrix. Okay, so we have here an illustration of a cartilage in the body, in the larynx, and in the trachea. Trachea or trachea, or whichever you pronounce that structure, larynx and trachea or trachea. So wherever we get the cartilage, either from the larynx or in the trachea, the appearance under the microscope will be the same. So it will contain perichondrium plus chondrocytes trapped in a lacuna. So here we have an illustration of a cartilage. So this is a hyaline cartilage wherein it is bound at both sides by perichondrium. So here, green and the brown color, it's just the stain used in staining the tissue. Then, types of cartilage, we go into the different types of cartilages in the body. The first one that is formed is the hyaline cartilage, the most characteristic type, wherein other types of cartilages in the body is just a modification of hyaline cartilage. So characteristics of hyaline cartilage, they're somewhat elastic, semi-transparent, with an opalescent bluish gray tint matrix that is in fresh preparation. And very important here is the matrix, which is considered to be a glassy appearance or homogeneous appearance. So we have here an illustration of a hyaline cartilage. So just look at the matrix or the ICS. So if there are no fibers that can be seen or the appearance is homogeneous, we know it is a hyaline cartilage with the territorial matrix area and intraterritorial area. So the same, we have a hyaline cartilage with a homogeneous ICS with the different isogenous group with the territorial and intraterritorial areas. So we have here, and again, an illustration of hyaline cartilages taken from the costal cartilage. So it will, wherever the cartilage are taken, it will give us the same morphological characteristics under the microscope with the chondrocyte trapped within a lacuna and the territorial and intraterritorial area of the tissue. So here we have again an illustration of a hyaline cartilage with an isogenous matrix and bounded by a perichondrium. Okay, so if we will go into the characteristics of hyaline cartilage, abundant chondrocytes, homogeneous matrix described as glassy appearance. Take note of this number 2, glassy appearance. And the reason why they appear glassy or transparent in appearance because the connective tissue fibers and the matrix may have the same or similar index of refraction. And take note for hyaline cartilage, presence of perichondrium or a dense fibrous outer covering. Then the hyaline cartilage is subclassified into a young cartilage. So we have the fetal hyaline cartilage and we have the adult cartilage and we have the articular cartilage. So 1 and 2, the hyaline and the fetal and the adult will just differ under the microscope by just looking at the sizes of the cell. So for a fetal hyaline cartilage, the cartilage cells are small, irregularly arranged chondrocytes with small and rare cell families or isogenous groups. But they will have a perichondrium which is very cellular in nature. Now, for the adult hyaline cartilage, the cartilage will contain larger or large spherical chondrocytes with also larger cell families at the center of the cartilage matrix. But take note, the adult hyaline cartilage will be covered entirely by perichondrium. So from a smaller chondrocytes in adult hyaline cartilage, the cells will become larger. Then we have the third type of hyaline cartilage. We have the articular hyaline cartilage. So this is mostly found in long bones of the body where joints meet. So the articular hyaline cartilage have the same characteristic as that of the adult hyaline cartilage. The only difference is that they have an incomplete perichondrium, or the perichondrium is absent over the articulating surfaces. From hyaline cartilage, the second type of cartilage is the elastic cartilage. Now, because of the fibers appear yellowish in fresh preparation, this is also referred to as yellowish cartilage. And the characteristic of the cartilage, it has a greater opacity, greater flexibility, and greater elasticity because of the elastic fibers. The elastic cartilage has limited distribution in the body. And with this type of cartilage, we can actually stretch out an elastic cartilage so it will be temporarily deformed without being structurally damaged. The protein making up the elastic fibers is referred to as elastin, and they are actually arranged in a meshwork formation. So here we have an illustration of an elastic cartilage. Take note, in the ICS you can see the elastic fibers that runs in all directions. So there is no proper direction of the elastic fiber running in all directions. So if we will go into the histological characteristics, abundant chondrocytes, presence of few cell families, abundant cartilage matrix, presence of abundant elastic fibers, and take note again, presence of a complete perichondrium. So what differs here will only be the number of the cell families, the presence of elastic fibers compared to the collagen fibers in hyaline cartilage, but the same characteristic presence of a complete perichondrium. So we have here an illustration of an elastic cartilage. Again, look at the ICS. You can see here plenty of fine fibers. We have the elastic fibers running in all directions. So an example in the body wherein we can find elastic cartilage is the external ear or pinna of the ear or in our epiglottis. So again, under the microscope, you can see the elastic fibers running in all directions. So this is an elastic cartilage 4D magnification. So you can see in between the ICS, the elastic fibers. So this can help us identify type of cartilage under the microscope. Then we have the third type of cartilage. We have the fibrocartilage. So it is a transition structure between dense fibrous connective tissue and hyaline cartilage. So in this type of cartilage, it has a scanty matrix because of the amount of collagen fibers present. Take note, the characteristics of the fibrocartilage, the chondrocytes or the cartilage cells are arranged in rows between bundles of collagen fibers. So take note of this. Okay, although the chondrocytes or the cartilage cells are scattered everywhere, but mostly they will be arranged in rows between bundles of collagen fibers. So here we have an illustration of a fibrocartilage wherein the chondrocytes are arranged in rows in between collagen or bundles of collagen fibers. Okay, so if we go into the characteristics of fibrocartilage, few small ovoid chondrocytes, so their sizes are smaller, rare and small cell families, again, very few cell families with scanty hyaline matrix. So the matrix is just few. It's because of the predominance of collagen fibers in bundles. And take note, number 5 characteristics, absence of perichondrium. So in the body, we can have the fibrocartilage as an example in the vertebral column or in between the intervertebral disc wherein we have a cartilage disc here and it is a fibrocartilage. So again, under the microscope, we can see the collagen fibers arranged in bundles of collagen fibers while the chondrocytes are arranged in. Let's go into the growth of cartilage. There are two ways wherein the cartilage will grow: the interstitial growth and the appositional growth. So it could be increase in the length of the cartilage or increase in the width of the cartilage. The only significant difference in the two processes is where they occur. No? So where they occur, it could be in the long bones, it could be in a flat bones. Okay, so they occur simultaneously and in a complete coordinated manner. So for the interstitial growth or endogenous growth, this will involve hyperplasia. So when we say hyperplasia, involving active mitosis, so there will be more cells that are formed and in making the cartilage grow in length. And this is dependent on the integrity of the cartilage. As long as the cartilage matrix is still soft and yielding, the cartilage will grow in length. So we have here an illustration of a chondrocyte undergoing cellular division, so mitosis, so more of the cells is formed. In mitosis, two daughter cells will be formed until new growth will be involved. So length of the cartilage will be involved in interstitial growth or endogenous growth involving hyperplasia. Now, the appositional growth or exogenous growth takes place at the periphery of the cartilage in the chondrogenic layer of the perichondrium. So in this process, there will be increase in the width of the cartilage, and the growth here will involve the chondrogenic layer of the perichondrium, wherein we have young cells here which will actually grow in size, so increasing the width of the cartilage. Okay, so that ends up our topic for cartilage.